Hello and welcome to the final episode of Odds and Ends Season 2. In this episode, we'll talk about the history of sugar beet breeding. The founder of the first sugar beet breeding program that we know of was one made by Franz Karl Achard, who started the project in 1802, and it started in a sugar factory in Lower Silesia. Due to the poor food quality of the sugar beet's primordial form, his attempts were viewed with skepticism, as all of the beets at that era were used for animal feed. However, the plant genetic material generated from this breeding program became the foundation stock for all existing sugar beet breeding programs. The selection efforts by Mr. Achard were derived from the large complex group of beet strains called Runkelrube. This complex had all the color types seen in beets today, including red, yellow, or white, alongside all the various shapes and sizes associated with beets. These traits are also found in modern forage beets that are grown today. Mr. Achard found that the specimens with white flesh had a high sugar content and began selecting seed from these white-fleshed variants. From there he found that the white-fleshed variants that did not grow out of the ground had higher levels of sugar. So from these white-fleshed populations, he selected only those that had not grown out of the ground for further reproduction. These plants became selected and were used for making seed stock. Through the work with Baron von Koppe and his son, he further developed the sugar factory and the plant line. The resulting open-pollinated line was called the White Silesian beet, which had a physiology and root yield nearly identical to our modern cultivars. Its sucrose content ranged between 7 to 10%, and according to the work of Dr. Snyder in 1939, it was likely an open-pollinated cultivar made up of low and high sucrose individuals. The breeding process was then taken up by Louis de Villemont, the next major contributor to the breeding program within sugar beet. Mr. Villerman was a member of a long line of seedsmen and plant breeders. He crafted the Villerman method of plant breeding, which was an improvement on the older breeding systems used for selecting varieties in sugar beet. The crude salt bath flotation method for finding roots with greater density was replaced by him in 1852 with the silver ingot method. In this method, the quality of the beetroot was determined by the weight, or as we call it, the specific gravity of its juice. A silver slug of known volume and weight was weighed when immersed in the juice, given an accurate amount of data to compute the specific gravity of the juice, which is used to indicate how much sugar was in the root. He also pioneered the use of a polariscope to determine sucrose levels within the sugar beet, and by 1860 these methods had become the standard for breeding sugar beet. In Europe, the breeding process was further improved in 1936 by Dr. Kuhns. This method involved preventing self-pollination and the selection of specific mother plants which were used to develop new seed. These specific lineages did not have any control crosses and instead required the use of appraisal and selection of progeny from the specific mother lineage. This was used to create new foundation stock and new cultivars, and this stock became used to further develop what would become the sugar beet industry within the United States. Much of the materials in Europe were sent to the United States, and various attempts were done to create a sugar beet industry within the United States. The first attempt was in Philadelphia in 1830, but the factory was shut down halfway through the building process. A second one happened in Massachusetts and was made in 1836. The factory ran for 2 years but closed down. The actual foundation for the long-lasting sugar beet industry within the United States was created in 1852 by the Mormons of Salt Lake. It was from there that these techniques spread across the United States, creating the standards we know today. The USDA started off by selecting various lines of European cultivars, but due to the weaknesses of these cultivars to the environment of the states, they were replaced by homegrown cultivars made by various people in a very decentralized way. In 1890, these homegrown cultivars were used to breed the new American cultivars, replacing the European stock altogether. These cultivars became the main source of genetics for all United States cultivars ever since. Between the years 1920 and 1940, disease began to run rampant in the United States, specifically the fungal diseases, black root and leaf spot. These diseases reduced yield in both the root size, the sugar content, as well as destroying specific plants within the stand. Other diseases such as beet curly top virus and insects like the beet leafhopper also made an appearance as well. Breeding for resistance to these two fungal diseases and this viral disease became issue number 1, and to deal with these threats, 2 doctors were selected, Dr. Kastner and Dr. Pack. These 2 breeders selected from the populations that had the most vigorous roots in the face of beet curly top virus, and these specimens were then crossbred with each other to derive new populations, and from these populations new cultivars were bred. Specifically, the cultivar US number 1. US number 1 was far superior in terms of resistance to beet curly top virus in comparison to the European cultivars that already existed at that time period. Another aspect was that mass production of seeds of this cultivar without selection was able to be done without reducing either agronomic qualities or losing any resistance to beet curly top virus. As time went on, several other selections from this population were made. Cultivars like US-22, US-33, and US-34, and several other derivatives were produced from selections of US number 1, and replaced their ancestor in 1938 due to their greater resistance to beet curly top virus. By the 40s, all testing of sugar beets from Europe was discontinued because it was deemed unnecessary. New cultivars were continually selected from these populations, as well as other populations made through other experiments. Such cultivars were US-15, US-58, and US-75. These strains were used to breed other varieties as well, and attempts to combine strains with different disease resistance were put into place. One such cultivar was US-216, which was bred between 1940 and 1941. This strain was not just resistant to the virus, but was also hyper-resistant to blackroot and really resistant to leaf spot as well. This strain was used to breed new inbred lines in F1 hybrids. And these are likewise more resistant to blackroot and leaf spot and the virus than most other cultivars in play. As a result, most of the cultivars of beet in the United States today are direct lineal descendants of this unique cultivar. Continued breeding in hopes of improving sugar content were made, and these were successful, as you can see by the screenshot above. The first attempt at improvement was made by Dr. Savitsky during his attempt to crossbreed sugar beet and Swiss chard whilst in the USSR. During that time, he found that primitive forms of Swiss chard present a great deal of genetic information, and through selective breeding with these primitive Swiss chard varieties and sugar beet, he was able to create new lines with higher sugar content than what it came before. This indicated that crossbreeding sugar beet and Swiss chard can create segregating forms with higher levels of sucrose than the original sugar bean parent, and that was happening in Europe. Whilst in the United States, Dr. Savitsky, not to be confused with the previous doctor, Dr. Savitsky, very similar names for different people, Dr. Savitsky did crossbreeding work with forage beet, and these crossbreeding experiments indicate that sugar lines derived from hybridization between forage beets and sugar beets can be created, and these lines have high levels of sugar in comparison to the previous pure-blooded sugar beet lines. In terms of disease resistance, Dr. Culbertson and Dr. Deming crossbred sugar beet with its wild ancestor, Beta martina, in 1925. These lineages showed high levels of resistance to leaf spot and other diseases such as curly top virus. These hybrids were tested in New Mexico, where it was confirmed that there's also some lineages that were resistant to both curly top virus and leaf spot. These hybrids were studied with great interest, but interest waned over time due to the fact that most of the lines have more wild traits than domesticated traits, making them ill-suited for production. The project was renewed in 1951 when Dr. Stewart revived the project and refined the breeding process, although no new cultivars have yet to be derived from this work. And that about covers everything. Thank you for watching.